Hello, my name is Patrick, and I'm going to start off with something that everyone hates. It's called audience participation. So I want you to put your hand up, please, if you've ever had the flu. Okay, that's a good, about 70%, yeah, almost all of you. That's great. So, well, it's not that great. So what's the big deal? Almost all of us, myself included, have had the flu, but we're all still standing here. We're all still alive. We've all made it to the other end. It was maybe a couple of unpleasant weeks in bed and a bit of a horrible time, but we're all still alive. So what's the big deal? Why even bother studying the flu? Well, the reason we were able to survive the flu is because we have a great, strong immune system. And unfortunately, not everyone is so lucky. People who are immunocompromised, people who have suffered cancer, they're extremely old, they're extremely young, they're really particularly sensitive to the flu, and it causes many deaths every year. And not to mention just humans, but we spend a lot of money every year vaccinating chickens, birds, fish, the lot against the flu. Billions of pounds and dollars are spent. And just think about the economic impact of half the people in your work being off every flu season for 2 weeks. I also have a bit of a personal grudge because the flu killed Mozart and I still hold a bit of a... So I've got a bit of a vendetta out. Now this is my favourite immune cell. This is called a dendritic cell. And what this cell is important for doing is raising the alarm, telling the immune system what to look for when it's looking for infection. And the flu, I like to think the flu as a little bit like Mr. Potato Head, because you get infected with the flu and your dendritic cell sees it and it says, that's what the flu looks like, I'm going to go and fight the infection against that. But the flu mutates so fast and so rapidly that by the next time the flu season comes along, it looks a little bit different. It's still a Mr. Potato Head, but it looks just a little bit different. So this cell's confused, it doesn't know what to do, it's not seen this before, so it has to go through the whole rigmarole again. And every time it changes, it gets a little bit different, and it gets sick again, and you have to get the vaccine again. And so every time it's a little bit different. So what people are trying to do now, they're thinking, well, instead of chasing after the little things that are changing every time with the flu, how about we go for the core, for the potato? And I think that's a really good idea. And one of the potatoes of the flu is this thing called NS1. And that's what I'm really interested in, because it's one thing that you find in almost all influenza viruses. So if we can figure out how to destroy that or antagonize that, then we can actually get to the bottom of this problem of the flu. So how does NS1 work? Well, this is a little happy cell, and it's been infected by some virus, and it releases all these signals saying, help, I'm infected, come help me. And so one of our helpful little dendritic cells comes along and helps, and then you get an immune response, and then you get better, and miraculously you're okay. But NS1's really sneaky because the flu comes in, it makes this NS1, and it kind of puts a gag over the cell. It stops it sending out all the signals, and so you don't get any response. And that's why you get sick for two or 3 weeks at a time, because it takes so long for your immune system to realize what's going on that by the time it figures out what's going on, you're already quite sick. So this is a really simple graph. I promise I'll keep it simple. And it just shows that as the progress of infection goes on, between 0 and 24 hours of infection, you get lots and lots of copies of NS1. And that's another reason that it's so important, because if the virus is prioritizing making it so much and using a lot of its effort and energy, then it must be an important thing. So we know it's there, but we don't know where, and we don't know really what it's doing. We can see that it's there, and we can see the effects of it, but we don't know the bit in the middle. It's a bit unclear. And I love this story. This is a squid. I think it's found in a jellyfish rather. It's found in the Galapagos Islands. And this is a great story about science for the sake of science, because this guy who just flew out there and went, I'm going to study these jellyfish, geez, and he found these amazing glow-in-the-dark jellyfish. And he found out that what was causing them to glow in the dark was this thing called GFP, which is green fluorescent protein. And he thought this was pretty cool, but he didn't realize that his discovery was going to revolutionize modern biology, because what you can do is you can take that same protein and you can put it into viruses, bacteria, and parasites, and you can actually see where they're going and see what they're doing in the cell. It's a little bit like if you had these glow-in-the-dark stickers when you were a little kid, and you put them on the wall if you're a bit afraid of the dark, and you leave the light on, and then at night time they glow up, and it's great. And that's exactly how this virus works. When you look at it down the microscope, you shine light on it, and it shines light back at you, and you can actually see them. And this is a perfect example of this. This is called a plaque assay. This is essentially a plate of cells which have been infected with the virus. And those little green dots you're seeing are lots of cells which have been infected by glow-in-the-dark green virus. And so this was proof to me that I'd actually been successful in making this virus and infecting my cells. So the first job I had was I had this big giant flask of cells which were all infected with virus, about half a litre. And I wanted to get a tiny, tiny amount, just about half a mil. So I use this process called ultracentrifugation, which definitely isn't as fun as it sounds. And what you do is you spin it really fast, and all the cells go to the bottom of the tube, and then you can actually get the virus, and you can get a tiny little half a mil vial which contains only the virus. And then what you do is you dump that virus onto cells, and then you can actually look and see what's going on. So you can see actually in real time what the virus is doing. And what I did was I used a thing called Confocal Microscopy, and what that is it's you look down the microscope and you can see your cells, and that's great because you've got a little 2D image of what your cells actually look like. But what you can also do is you can take images above the cell and below them and smush them all together and make a nice 3D image. And then what you can do is you can take that and you can animate it using this software called Damaris. Now these are uninfected cells, and the blue dots you see are the nucleus, the control center of the cell, and the red is the cell wall, which is like a sort of the outer limits of the cell. And these are uninfected, but this is just to show you that what happens is we take every time you see this blue color, it makes a nice surface and you can see the nucleus, and the same goes for the red. And then what we did is we infected some cells and did the exact same thing. So this time you can see the nice blue nucleus and the cell membrane with the red, but those little green dots you see are actually the virus that's infected the cell. And then what you can do is you can go right down to the surface of the cell and pan through, and you can see, well, is that actually inside the cell? Is it outside the cell? Where is it inside the cell, if it is inside the cell? And that's really important because that answers that question of what it's actually doing, and we can understand how much of it's being made, where it's being made, and where it's actually being used. And the one that's my favorite is we did these single cell imaging as well. So you take a single cell out of the whole plate, you look at it on its own, and you do the exact same thing. And then you can actually look and see, well, you can see a couple of those green dots are sitting on the surface, some of them are inside. And then once again, you pan through the cell, and you can actually see what it looks like on the inside. So this is really important because this allows the development of totally new treatments, and more importantly, tailoring of the treatments that we have today. Because now that we have a greater understanding, we can design treatments against it. Because the thing is, if you remember back to what I was saying about the NS1 shutting up the immune system, well, that's the reason you get sick, is that it takes a long time before your immune system cottons on to you being infected. And then by the time you've been infected, you're filled with the virus and you get really sick. But if we can create some kind of drug target or something to go against this NS1, then we can actually get you feeling better before you've even been infected. Or you can put something against the NS1 into the vaccine so that as soon as your body sees this NS1, it goes, that's a virus, let's go and kill it. And then that would solve the whole vaccine problem after all. So that's my research, and I think it's really interesting. I think that this kind of research, rather than researching the things that change about the virus, are going to be what's the next step. And I just want to say a very quick thank you to the lovely people at the CVR for helping me out. Ed sort of led me through kicking and screaming all the way to the end. My favourite thing about this conference is that it gives me the chance to talk to non-scientists about my research. And I'd love to know a little bit about what you guys think is good and what you guys think is bad. And I would absolutely love to answer your questions. So thank you very much.
So I have a question from somebody in the audience who has asked, how much easier does this make the development of a treatment? What has to come next now?
So a lot of the treatments out there aren't actually targeting NS1 at all. So it's something that, the name NS1 actually means non-structural one. It was thought to be this kind of useless it wasn't involved in the virus at all. And very recently, the guy on the far left there showed that it actually had this function. And so the next step is going to be figuring out, so I've showed quite nicely that it can be inside cells and it can be outside cells. But the next step is to also label other things in the cell to see where it's going. So if it's maybe going into other machinery in the cell, and then you can understand better how to attack it. So that's the next step.
Okay. Have you done any structural studies on the protein, and do you know exactly how it functions?
So the NS1 or the GFP? Or a bit of both? So what has actually been shown by other researchers is that, so they're roughly about the same size, NS1 and GFP, and when you put them together, it actually kind of makes the NS1 work more poorly. It's not as effective at what it does when it's got this GFP attached to it, which is not super important when you're doing something like this, which is just kind of seeing where it's going. But if you wanted to actually understand how it was functioning as taking down the immune system, you would maybe struggle to use a GFP tagged one because it's going to lose some of its strength, if you like. Yeah.
By that, I meant, do you know, or do you have any leads as to how it is involved in immunity?
Oh, absolutely. So there's about 5 or 6 different completely solved mechanisms. If you're a particular molecular biology geek, it inhibits PI3K, which is involved in cell death. So what happens is when you get a lot of viruses infecting a cell, one of the defense mechanisms is the cell just goes, I'm in trouble, I'm going to die. And it sends out the signal called PI3K, and this NS1 inhibits that and it stops it from dying. So again, it just keeps everything on the down low and stops the immune system catching up with it. So there's like more than I could go on about for days and days.
You know how usually you have viruses which evolve over time, it makes it even harder for us to target the drugs. So I was just wondering, does this NS1 ever change or evolve throughout time, or does it not?
It seems to be very conserved in that it doesn't change. The thing is, the flu naturally mutates a lot, so what happens is maybe half of the population would lose its NS1, and then it wouldn't actually be able to infect cells properly, so that would just die out, and the ones that survive live on. So it seems that because it's one of the very few things that's found in almost all the influenza viruses, so that would tell us that it's so important that it's sort of essential to life. So it's very unlikely that it's ever going to be lost or changed. The only way it would be changed if some mutation happened to make it better, and then that would be the sort of the magnum opus, and it would continue on in that direction.
Okay, well, see if we return. Thanks very much. If you'll join me again, it's fantastic.
Thank you. Thank you.